So today we are switching over to look at adaptive immunology, and we're going to be looking at it in the context of Guillain-Barré syndrome. One of the unique things that you'll find out about immunologists is we like to name our diseases after ourselves. There's a bit of a vanity there, and so this is a disease characterized by two French physicians, and so this right here is the jawless hagfish, and what's unique about this fish is that it has no adaptive immunity. It only operates with an innate immune system, and part of its defense mechanism is to release these slime glands. As if it wasn't ugly enough, it releases these weird slime glands to try and fend off its invaders. And part of that is triggered by its pathogen recognition receptors. And so its immunity is driven primarily by its innate immune system, and this is part of its defense. So, Guillain-Barré syndrome. It is an acute autoimmune polyneuropathy. Polyneuropathy means that it is a multifaceted neurological disease, and it's one of the most common forms of flaccid paralysis. And I think you guys talked about flaccid paralysis in your botulism lecture. Some of these symptoms: you suffer from pins and needles and nerve pain. In addition, there's severe muscle pain and contractions as a result of losing control of muscle tone, and furthermore, there's unsteady walking, or you lose the ability to walk. You lose the ability to walk and have fine motor skills, and you actually lose fine eye movement, facial movement, speaking, chewing, and swallowing, and you largely lose your autonomic dysfunction. So autonomic means the functions of your body that you're not actively controlling. So your heart rate, your blood pressure, your body temp, your bladder, and lastly, many patients will die as a result of losing the ability to breathe or lose the ability to control their breathing. The progression of the disease. It is triggered by an acute infection that kicks it off, and then after the course of a month, that's how it takes the course of a month for the disease to progress, and it plateaus after a few days. And so even though the disease takes the course of a month to be initiated, to progress, and to plateau, the resolution of this disease, for it to go away, can be on the order of months to years. And for some patients, after a few years, they still suffer from weakness. So doctors will oftentimes have a hard time distinguishing this disease from other polyneuropathies, and that's characterized as differential diagnosis. And so, as you guys discussed with botulism, there's a really similar presentation as it is with GBS. That's also true with heavy metal intoxication. So if you have lead poisoning, it'll present itself fairly similar. And furthermore, myasthenia gravis is another neurological disease that you'll suffer from autonomic dysfunction and a lot of difficulty with walking. And lastly, that is also true with polio and tick bites. So the etiology of this disease. Guillain-Barré syndrome is often caused by a bacterial infection, and it's most often caused by the bacterium Campylobacter jejuni, or C. jejuni. And the way the pathogenesis works is it's an autoimmune disease. As I understand, you guys covered autoimmunity to some degree already. And autoimmune disease is largely where your body recognizes the self as foreign. It attacks whatever presented protein your immune system recognizes as foreign. And what happens here is your body will recognize your Schwann cells as foreign. Schwann cells are peripheral nerve cells that help provide the encasement around your nerves. That encasement, that sheath, is called myelin. And so in your peripheral nerves, Schwann cells are the myelin-producing cell in your peripheral nervous system. There's a different cell in the central nervous system, but what's critical here is that I mentioned earlier that the symptoms include difficulty walking, difficulty breathing, difficulty with eye and facial movement. So your nerves in your peripheral nervous system, that conduction across the nerve is facilitated by the myelin your Schwann cells are making, and your body is attacking your Schwann cells, and GBS. So when we discussed last class, we discussed primarily on pathogens or specific antigens, but not about cells being attacked. And so in autoimmune disease, a lot of times your cells can be attacked, just similar with type 1 diabetes. In type 1 diabetes, your beta cells, the insulin-producing cells, are what's being attacked. Type 1 diabetes is an autoimmune disease. And so here, your Schwann cells are attacked, and as a result of that, myelin is degrading, and the ability to sufficiently excite your peripheral neurons is lost. So before we move any further, the thing with immunology is that it's really difficult to try and follow some of the different terms if you're not familiar with them, or sometimes people use different terms to mean different things. And so we're just going to kind of cover some of the ground-based terms here. And so as I mentioned earlier, autoimmune disease is where your body recognizes the self as foreign. Ideally, you want your body to recognize self as self and not attack it. But in the case of autoimmune disease, with different autoimmune diseases, different parts of self are recognized as foreign. And what happens is the self that's recognized becomes the antigen. An antigen is a substance that's capable of inducing an immune response. And so whenever we talk about immune cell activation, usually your immune cells, they're not incredibly active. So they're oftentimes moving around and kind of patrolling, but you need some sort of antigen to activate. And in this case, antigen is whatever activates the immune cell. And in autoimmune disease, that antigen is an autoantigen. That's self-antigen. And so whenever we talk about the antigen in autoimmune disease, we refer to it as autoantigen. And so what's important is that whenever we've talked about what activates your pattern recognition receptors, or what we'll talk about later on today about antibodies, there's a specific portion of an antigen that we're interested in, and that's called the epitope. And so the epitope is the specific part of an antigen that elicits the immune response. And so what ends up happening is every antigen will have many different epitopes on the surface of it. And so whenever we talk about antigen and epitopes, an antigen has many epitopes. But as we'll talk about later today, an antibody will bind one specific epitope. And we'll talk about that a little bit more. So an antibody is a protein that binds to a specific epitope. All right, so antibodies are incredibly specific to the extent that an antibody will recognize a specific epitope. And next off, we'll talk about anaphylaxis. So anaphylaxis, some of you guys may have heard that term thrown around with anaphylactic shock. Whenever you hear movies, they'll scream, "He's going into anaphylactic shock." Anaphylaxis is a systemic allergic reaction. So sometimes you'll have a localized allergic reaction where if you're allergic to some sort of fabric, or for example, if you're allergic to latex, if you're wearing latex gloves, you'll have a localized allergic reaction in your hand. But a lot of times, in response to really severe allergic reactions, to example, bee venom, you'll have you'll have an anaphylactic response. And so I'll discuss later what that anaphylactic response will look like and how that happens. Next up is proliferate. And so this is distinct from cell growth. Cell growth is oftentimes indicative of cells getting larger, but proliferating means you're getting more cells. You're getting an increase in cell number. Bless you. So proliferation is with respect to the cell number. You're getting more division and more cells. And that's going to be very important later on when we talk about a specific cell of the adaptive immune system. And lastly, differentiation. That is an important process in immunology. In your pre-lecture, you guys learned about B cells and plasma cells. B cells are the precursor to plasma cells. They'll differentiate and become a more specialized cell called plasma cells that make antibodies. With the innate immune system, we talk about monocytes becoming macrophages when they leave the bloodstream and go into the peripheral tissue. So monocytes differentiate into macrophages, and they'll oftentimes differentiate into a special flavor macrophages depending on what tissue they go to. So we're going to talk about the specific qualities of the immune system, the rare qualities of the immune system, with the adaptive, the arm of the adaptive immune system. I guess I should say first, based on what we just discussed, what questions do you guys have? Not if, what questions do you guys have? All right. So with adaptive immunity, it is incredibly diverse. So we discussed with innate immunity that there's categories of antigens that your body can respond to, but with the adaptive immune system, it is incredibly diverse, on the order of trillions of antigens that they can recognize and respond to. And so with innate immunity, we discussed how we are born with all of these different pattern recognition receptors, but in adaptive immunity, we develop antibodies in response to antigen, and we develop cells over the course of hematopoiesis, as you guys discussed in your pre-lecture, that we can have T cells and B cells that respond to a plethora of different antigens. And in response to that, it is incredibly more specific. You're able to respond to very, very specific epitopes. We discussed how you can respond to a specific PAMP, and that's, an entire biomolecule. But here in adaptive immunity, we're talking about several amino acids that your adaptive immune response can respond to. And lastly, memory. With innate immune system, we talked about how over the course of evolution, we've developed and remembered these pattern recognition receptors and the ability to respond to different PAMPs that elicit disease. But here in adaptive immunity, we developed this over the course of a lifetime. And so for example, for colds, we have, I think, 250 different kinds of cold strains out there, but you only get it once in your life, that one strain. And so after you get it once, you're never going to get that one again, because your body is tuned to respond to that specific, to remember that specific virus strain that you don't want to ever get again. And so you acquire this immunity. So adaptive is also referred to as acquired immunity. So you acquire this immunity and you remember it for the course of your lifetime. So the divisions of the adaptive immune response are pretty similar to the innate immune system. So we're going to look at it with respect to humoral immunity and then cell-mediated immunity. So with the adaptive immune response, the humoral immunity is primarily comprised of your antibodies. And so we're going to talk about what antibodies are, how they work, and why they're important. And the cell-mediated adaptive immune response is primarily carried out by T cells, but B cells are also important in that response as well. So we have two types of lymphocytes, as your pre-lecture stated and as I've been referring to. You have your B cells. So your B cells, though it might be hard to remember the distinction between the two, but your B cells are important for antibody production. And as we mentioned earlier, they differentiate. So B cells differentiate into plasma cells. And once they become plasma cells, they can dump out a ton of antibodies to attack whatever pathogen that your body's infected with. And what's important is that these antibodies can help facilitate. They can facilitate the response to whatever pathogen, whatever antigen is present. And they can isolate it. They can help promote the engulfment and digestion of this antigen. And what's important next is that they can help lead to the presentation of this antigen. And so, for example, if we have a B cell here, they have every B cell. So we have lots of different B cells and T cells within our body, and each B cell will make an antibody that recognizes one epitope. Only one epitope. They'll make a ton of that antibody, and it'll be on the surface of a B cell. That was bad. So this Y is a poor man's antibody on the surface of a B cell. You'll have many of them, but it'll recognize one epitope. And when you have an antigen that comes in and binds here to this antibody, what will happen is it'll engulf it, it'll digest it, and then it'll present it out here. So it'll bring it in and then present it out here. And This is a weird diagram with no letters at all. I'm sorry. And so this process is called antigen presentation, and we're gonna understand this a little bit more as we go on. Next, your T cells are critical for cell-mediated immunity, and so they're really critical. The reason why they're critical in cell-mediated immunity is that they'll actually go out and directly be in contact or touch cells that they're targeting. And so there's different proteins on the surface of T cells and the cells that they're targeting that they'll touch them and then they'll tell them to die. And that process is called that death process is apoptosis. It's best characterized as programmed cell death. So what they'll do is if they have a cell that needs to die, that shouldn't be around, they'll touch it and give it a signal to die, and it'll just elegantly die off. Last lecture, I think I said that innate immunity is fast but dumb. Adaptive immunity is slow but smart. And it's smart in the sense that it can go off and carefully touch many different cells, and if a cell needs to die, they'll just give an apoptosis signal to tell that cell to die, and it'll die. And lastly, T cells will secrete a lot of different proteins that can modulate different cells in the immune system and their responses. And so this cell, the T cell is really critical for mediating cell-to-cell immunity. And that's why HIV/AIDS is so devastating, because they target, every virus has a specific target that they're trying to infect and kill. With HIV, they're targeting T cells. And so you lose this ability to have this careful cell-to-cell mediated immunity and adaptive immunity. Any questions? You guys must be, there we go, we got a question. Yes, I'll actually go into a little bit more careful detail later on, but, and so I'll touch back to it when I'm able to discuss a little bit more about what's happening here and what's happening here. So throughout our immune systems, we have antigen receptors, and so we talked about how in innate immunity we had the antigen was largely PAMPs, and we had pathogen recognition receptors, and those were the antigen receptors we were really interested in. But here in adaptive immunity, we have different kinds of antigen receptors we're going to be focusing on, and so the antigen receptors are the way B cells and T cells will recognize their antigens. And so for B cells here, the antibodies on the surface here are going to be recognizing the antigens. And as you'd imagine, an antigen receptor is a receptor whose ligand, what it binds to, is an antigen. And so immune cells will express one and only one antigen receptor, one kind of antigen receptor. And antibodies are one class of antigen receptor. So for B cells, their antigen receptor is an actual antibody on the surface of it. And this is actually called the B cell receptor. I'll say BCR for B cell receptor. So on the surface of a B cell, there's an antibody that will recognize one and only one epitope. And that antibody is the actual B cell receptor. And so this ability for B cells to recognize one and only one epitope is how there's what's called cognate. They'll encounter a specific cognate antigen. And so it's a matching antigen where the antigen on the surface, or the epitope on the antigen, will match the antibody on that specific B cell. So we have many, within our lymph nodes, we have lots of different kinds of B cells and T cells that recognize different epitopes. And what will end up happening is when you have an infection, the antigen and another cell, the dendritic cell, will drain to your lymph node. And what will end up happening is, and we'll talk about this later, is dendritic cells will just go around and just touch different T cells and say, is this the epitope that you recognize? Is this the epitope that you recognize? And B cells will go around and just monitor different antigens. And when they bind that antigen, they're activated. Now there's a second step necessary, but that's outside of our scope. But what's important to know is that each B cell has an antibody that recognizes one and only one epitope, but it has many of them on the surface. So antibodies are a tetrapeptide structure. And so you have 2 light chains and 2 heavy chains, and they're identical. So I hope this makes sense. Let's see. So over here you have, let's see. So this right here, can you guys see that okay? All right. So this inside long arm is called your heavy chain. And then this little short arm right here, that's your light chain. I'll say light chain for LC for light chain. And then this long arm is your heavy chain. Now, it's hard to appreciate here in this image, but in this image, what you'll actually see is this entire yellow. I'm also colorblind, so if it's not yellow, please don't judge me. This yellow long arm is the heavy chain, and this short white arm is your light chain. All right? And what's important to know is that on both sides of your antibody, you have 2 light chains and 2 heavy chains. They're the same. All right? You have the same light chains and heavy chains on both sides of your antibody. And what's important to know is that there are also variable and constant regions. And so the variable regions constitute your antigen binding site. And so I don't know if you noticed, I have these little breaks here in the antibody. So these breaks are important for your variable and constant regions. And so here you have your variable regions, and here you have your constant regions. And so because you have these light chains and heavy chains, and it's a tetrapeptide, there's a lot of different ways that people break up antibodies, and it can get a little confusing. And so if something's unclear, again, please raise your hand, and I'd be happy to try and clarify. Yes? So what these antibodies recognize epitopes. And so I talked about how this variable region here is important because this is your antigen binding site, FAB. All right, so AB is antigen binding. So this is where this is this part and this part. So there, again, you have the same light chain and heavy chain on both sides. So these are identical. These are the same. All right, and this is where your antibody will bind an epitope. Does that make sense? Yeah, go ahead. Great question. So what'll end up happening is, I'll touch on this later, but in immunity, you'll have what's called cross-linking. So this, for example, I'll just draw a weird oblong thing here. All right? So it's binding to one epitope here, and if you have the same antibody over here, it'll bind to the same epitope, the same exact epitope, but over here. Like if it's a matching epitope, you'll have one antibody that can bind 2 antigens if it has multiple epitopes that are the same. Does that make sense? So here. So for example, all right, so I should have used this. I have 2 antibodies here. So if I have, this is another antigen right here. So the way antibodies will work is that if you have a ton of these antigens within your body, and what your body will do is if it has antibodies that recognize that pathogen, and it'll recognize that pathogen by touching the epitope, by saying, okay, this has an epitope that I'm made to recognize. So I'm going to bind it, and then you can have 2 antibodies that bind 1 antigen. You can have multiple antibodies that bind 1 antigen, as long as that antigen has multiple epitopes for it to bind. Does that make sense? Okay. Yes. Yes. Yeah. So, for example, in my really crudely drawn image, this one antibody is binding to 1 antigen here and 1 antigen here. As long as they have the epitope that this antibody is made to recognize, it can bind 2. Yes, Dr. James.
I just want to provide a clarification.
Uh-oh.
There's a lot of terminology. I just want to make it clear. The instances that Dan are referring to, let's say you have a bacterium, right? It's in your bloodstream, and Dan already talked about the complement pathway antibody mediated. We're talking about the same antibody. antibacterial, right? Is it recognizing the bacterium because it's a bacterium that coats all the surface? No. All right. What happens is the bacterium has hundreds of thousands of molecules, epitopes, identical epitopes, because it corresponds to some protein or sugar or other type of foreign epitope on the surface. Identical, 40, 50, 50,000, 5 million times on the surface. So I want to emphasize that this multiplicity in binding, if you're talking about one type of antibody, occurs only if the exact same epitope is repeated on a pathogen surface. Okay? There are other ways in which you could get the same protein, or one antigen with multiple epitopes coded by many different antibodies, each recognizing different parts of the epitope, each recognizing different parts of the antigen. But that's many antibodies to many epitopes mapping. Right? So the thing that you need to take home is that each antigen, they have many epitopes. Each antibody recognizes only one epitope, and it's a combination of certain pathogens having many of the same epitopes, or the immune system generating many different antibodies that can recognize many different epitopes on antigens.
Thank you. I think I saw another hand that went up. Yes.
For the variable region, does both the heavy and the light chain participate binding, or is only one or the other?
Both participate. Any other questions? All right. And so we have the antigen binding domain, and we also have an FC domain, the leg. And so the FC, you can think of it as like a cell binding domain. And so the FC domain, I'll draw another antibody over here. This down here is the FC domain, all right? And so this is what binds to the surface of different cells that express an FC receptor, all right? And so, antibodies are really elegant in that they can serve many different purposes between, isolating a pathogen or binding to the surface of a cell, and when it recognizes it, it can elicit a very specific response. And so this is going to be really key later on when we talk about FC receptors. So as we discussed a second ago, we have an antigen binding site. So you can think of it as almost like the way it was discussed when I first took immunology is that it's almost like these two Dixie cups that kind of sit on, you go over here, where you have these like little loops. You can think of them as like little cups or loops that are primed to recognize very specific peptides. And so, as was asked earlier, both the heavy chain and the light chain are a part of the FAB region, your antigen binding site. And so this constitutes the variable domain because, and I think in a few lectures you guys will talk about antibody diversity, but you have both the light chain and the heavy chain that can be switched out to create really diverse antibodies that recognize different peptides. And these little cups or loops can also be rearranged to recognize different epitopes. And so between differences in the heavy chain and the light chain and differences within these loops, you can create an incredibly diverse antigen binding site on the order of trillions. And so what can end up happening is you can create these immune complexes. And so thankfully we kind of have that drawn up here on the board a little bit. But antibodies are multivalent. They can bind to multiple antigens. So one antibody, as drawn here, one antibody can bind two antigens. And they can form what's called an immune complex, and they can cross-link. So for example, on the surface of mast cells, they'll recognize a specific flavor of antibodies. And the only way a mast cell will become activated, sorry, that's a terrible thing to say. One of the key ways a mast cell can be activated for an allergic response is when two antibodies bind to one antigen and they're cross-linked. What ends up happening is that the FC receptors, the receptors on the surface of a mast cell that bind this this FC region, they'll cluster together and prompt mast cell activation. And so this is true in a lot of different ways. And so as is illustrated in the diagram here, you have 3 antibodies that recognize a hexagon antigen. Now, each antibody will recognize a very specific epitope, but one antibody can bind 2 antigens. And when you have multiple antibodies together binding antigens, you'll have an immune complex. And if this is occurring on the surface of a cell, they'll cross-link, and your FC receptors will get closer together and activate the cell. And so this can also be helpful in the case of pathogens inside your bloodstream, where if you have multiple antibodies recognizing different epitopes on the surface of this pathogen, it can precipitate out whatever that pathogen is. And so we use this a lot of times in the lab as well. Yes? That's one of the ways. So this is what Dr. James was touching on earlier. So on one antigen, so for example, let's say, so I'm going to use the illustration on the slide as an example. Let's say, for example, this antigen is not the same on this arm here. So this antibody 1, recognizes that specific arm of this antigen. Oh no. All right. So let's say, for example, on the top 2 antibodies in this image, the top antibody only recognizes that specific arm of that hexagon antigen. It is made to recognize only that one specific portion of that antigen, that epitope on that arm. But the antibody 2, the second one down, is made to recognize that arm. And so you can create immune complexes with antibodies that recognize different parts, or sometimes it can be, or in other cases, if the epitope is the same on both sides, it can be the same antibody. But what they'll do is they'll form these complexes that will isolate and eradicate the antigen. Are there any other questions? Yes. So there's a lot of different ways. We'll touch on a few of them, but Dr. James referenced complement. If you have multiple antibodies if you have multiple antibodies binding to the surface of a bacterium, and this is occurring in the circulation, you can have the complement proteins come in, and the complement proteins will be the classical pathway. I'm sure you guys are experts, as we discussed this last lecture. The classical pathway reacts to antibodies binding to one specific complement protein, C1q. And so the classical pathway is going to be activated in response to multiple antibodies binding to the surface of bacterium. The size of the antigen on this slide is not going to correspond necessarily to the actual size of the bacterium relative to the antibody. So it's not always, the antibody is not always going to be this much bigger than the antigen it's recognizing. Another way that antibodies can eradicate a pathogen is that, as was mentioned earlier, the B cell receptor is an antibody. The B cell receptor will bind to an antigen, recognize it, engulf it, and then present it out for other cells to try and recognize it and facilitate eliminating that pathogen. Does that make sense? Any other questions?
The one other one that I would just add is probably going to be coming on the slide. It's called neutralization. Imagine that the hexagon on the slide is a virus. There in fact are icosahedral viruses that have that type of shape. Repeating units all the way around, and they form a geometric shape. And those repeating units are critical for the virus to dock upon a host cell. They get recognized at the cell surface, internalized, and detect. If antibodies are coding all of those surface proteins, it creates a steric barrier for that virus to go in and infect the host cell. It can't interact with the proteins on the host cell surface. So that's separate from the complement pathway, separate from the internalization and antigen presentation pathway that Dan talked about, but another means by which you can dampen down an infection or adverse host cell response by antibodies.
So as we discussed a second ago, oh, yes. So a pathogen, I mean, largely there's not a significant difference. An antigen is whatever, an antigen is a substance that elicits an immune response, and oftentimes a pathogen is an antigen. You'll have pathogens that invade and will induce an immune response. And antigen is kind of antigens are the terms that we use that, as a general term, we use to refer to things that activate the immune system. Now, I think, Dr. James, I might lean on you for this. Not all antigens would necessarily be characterized as a pathogen.
Peanut allergy is antigenic for some people, but it doesn't cause a disease.
Right. It's not an invading organism.
Pollen, that's very antigenic. Pathogens cause disease. Okay? And so that's where you would think the distinction.
Okay. Thank you. So the way these immune complexes work is that they'll facilitate, as we discussed earlier, complement activation, specifically with classical pathway. They can induce phagocytosis with macrophages and neutrophils. And so there's specific flavors of antibodies that can bind to the surface of macrophages and neutrophils, and that'll facilitate the engulfment or phagocytosis of specific antigens that those antibodies recognize. And lastly, degranulation of granulocytes. So I worked in a mast cell lab, and I mentioned earlier that people have their specific cells or things that they care about in immunology. The people I worked with think mast cells are the best thing in the world. Most of the world might not necessarily agree with them, but degranulation is really unique and important because specifically in the context of mast cells, those are the cells that are responsible for allergies. And a lot of times activation of cells and the things that happen downstream of that are on the orders of hours. Degranulation can happen on the orders of minutes. So a lot of times when people have an allergic reaction to something, in a matter of 5 minutes, their face is puffy, they have hives. This degranulation occurs rapidly and really quickly, and it involves an emptying of all the granules that you guys have seen on your pre-lecture slides, where there's these electron micrographs, these like gray and black images of cells that we look at sometimes. The granulocytes have these like little pockets that are filled with cytotoxins and proteins and lipids and histamine in the case of mast cells. And so they'll dump out a lot of histamines, and that happens in response to immune complexes on the surface of these cells. So activation of non-lymphocyte white cells. So again, antibodies bind to antigens. We have antibodies recognize specific epitopes on these antigens. An antigen might have different epitopes, but it'll have many epitopes, but the antibody will recognize only one specific epitope on that antigen. And these complexes bind to Fc receptors. And so I mentioned earlier that this region here is called the Fc region. And you'll have a cell that has a specific Fc receptor. And so when this antibody is binding to the cell, it's not going to bind. It shouldn't be binding with the antigen binding site, because that's how the antibodies will recognize a specific epitope of an antigen. But rather, it'll dock and bind on this Fc receptor, and cells will have many Fc receptors, and there's lots of different kinds of Fc receptors, and it'll vary from cell to cell. And what's unique is that the Fc receptor of different antibodies will kind of dictate what the outcome is of the antibodies becoming of binding to a antigen and becoming activated, or the cell being activated. And so this is most notably with granulocytes. Oh, yes. So in some cases, yes. It'll depend from cell to cell. So for example, just because this is the context that I came from. For example, mast cells throughout your body is covered with antibodies already. All of our mast cells already have a specific antibody on them. and they're just waiting to recognize specific epitopes on different antigens. But in different cases, your antibodies will already, in some cases, will already form an immune complex with some antigen. And when your cells, because your cells have, I just drew only one Fc receptor. There are thousands of Fc receptors and different kinds of Fc receptors on different cells. And if there's already an immune complex, if it docks and binds to the Fc receptors on that cell, that can also trigger activation. Does that make sense? Any other questions? So when we actually get to the next slide, we're going to actually talk about different kinds of antibodies, and I'll kind of say what the corresponding Fc receptors are. And so with the complexes that bind, the immune complexes that sometimes bind to the Fc receptors, or antibodies that are on the Fc receptors and then bind to an antigen, that'll activate that immune cell, and it'll induce degranulation or phagocytosis. And so in this illustration, this is an instance where you already have an antigen, an antibody-bound antigen. So this antibody is made to recognize the specific epitope on that antigen. It'll bind to the Fc receptor, and it'll elicit activation and a lot of different downstream outcomes. And so with different cells, you'll have different outcomes. With mast cells, you'll get asthma and allergic responses because of histamine release. It'll degranulate histamine, and that'll induce severe asthma or allergies. In other contexts, with neutrophils or macrophages, you'll get different proteins released, different lipids released, different enzymes or proteases released that are meant to attack whatever that antigen is. So we have 5 main flavors of antibodies. The antibodies, the nomenclature we use is IG blank. And IG stands for immunoglobulin. And that immunoglobulin refers to the structure of the antibodies. And so the first noteworthy antibody is IgG. And so IgG is the most abundant antibody. And there's actually, this is not important, there's actually also like 4 different kinds of IgG antibodies. And so it's so abundant that it constitutes about 20% of the protein in our serum. Next is IgD. That's an early antigen receptor on B cells. So as I mentioned earlier, B cells have antibodies on the surface. Those are the B cell receptors. And an early form of that B cell receptor is usually IgD. Next up is IgM. IgM is the class of antibody that is usually found on the surface of B cells as the B cell receptor. And it's also the first class of antibody made in response to an antigen. So when your body is first making antibodies in response to an antigen, it's first going to make IgM, but in subsequent exposures, it's going to make different classes. IgA is the class of antibody that's important for mucosal immunity. It's also, I believe, the class of antibody that is passed from mother to child during breastfeeding. So it's the critical antibody that's important in the mucosal epithelium. Lastly, for any mast cell fans out there, IgE is the critical class of antibody that's important for the allergic response or asthma. And so when thinking about which antibody, like how much of which antibody is most abundant, the acronym that I was taught is GMADE, G-M-A-D-E. And so again, IgG is the most abundant. Again, and then after that, IgM, because IgM is the first flavor or class of antibody made in response to an antigen. And then following that, IgA and IgD and IgE. So GMADE. For the mast cell biologists, IgE is the least abundant, but they also like to say it's the one that can kill you. So despite the low abundance of some of these antibodies, they still all serve different and important functions. Do you guys have any questions? Yes? To the best of my knowledge, it's usually not made on secondary exposure. But Dr. James, that's my understanding. What's your answer?
The lecture after that coming one, Professor Parker talks about at the molecular level how the immunoglobulin genes are regulated and synthesized and developed over time. There's a very discrete transition from one antibody class to another antibody class. However, I don't think it lines up so clean with, like, do they see it the first time and then do they see it the second time and it changes over? That what determines it is the strength of that interaction. And so you can envision scenarios where there are many weak interactions over time and then eventually we get there and go over. Or if a particular B cell encounters a really strong non-self antigen early on, maybe that first trigger is enough to go and begin expressing the other classes of antibodies. But what is true is that you always start with M, and then you'll go to D, and then the individual B cells will diverge on different paths.
Are there any other questions? All right. So different antibodies. Oh, and I forgot to mention this. So with I mentioned, we talked about the 5 subclasses. The corresponding FC receptors usually have FC followed by the Greek letter of that class of antibodies. So the FC receptor for IgE is FC epsilon. So between all of you being engineers, taking calculus, physics, and a large portion, I imagine, are in the Greek system, I imagine all of you really know your Greek alphabet really well. So for example, IgG binds to FC gamma. So each antibody will bind to an FC Greek letter corresponding receptor. And those FC receptors, so FC gamma will have a different signaling pathway than FC epsilon. Does that make sense? All right. So each antibody class will bind to antigens a little bit differently. So, for example, IgM can form a pentameric structure where different antibodies will come together and bind and form this almost like neutralizing structure around an antigen, whereas IgG will form a dimeric structure when binding to different antigens. And so antibodies can come, you can come across antibodies in either a cell-bound form or a secreted form. The secreted form is going to be in your plasma, and that's what's going to be binding to the FC receptors on different cells. So again, IgE will bind to FC epsilon, IgG will bind to FC gamma, IgD to FC delta, what am I missing? IgA to FC alpha, IgM to FC mu. And so all these different secreted antibodies will bind to those different FC receptors. The bound form will occur with respect to the FC gamma the FC portion being present on the surface of the cell. And so, as I mentioned here, the B cell receptor is an antibody. And again, as we talked earlier, it'll recognize a specific epitope, but you'll have tons of antibodies across the surface of this B cell. And you'll have many different B cells in your body that will recognize different pathogens. But again, one B cell receptor, the B cell receptor is an antibody. So one B cell receptor will recognize one specific epitope on an antigen. And so the two types of lymphocytes that we're going to be focusing on are the B cells and T cells again. So B cells are critical for the humoral immunity because they'll be the antibody producing cell that's critical for adaptive humoral immunity, and T cells are critical for cell-mediated immunity. And so what's critical about B cells is that the B cell receptor on the surface of B cells will bind free antigens. That's one of the critical roles for B cells in immunity, in that in response to, in addition to making antibodies, they're also critical for binding free antigens that can drain to the lymph node through your lymphatics, and your B cells will kind of go through and touch that antigen and try and recognize it. T cells have the antigen receptor, but it's not quite the same as a B cell receptor. So the B cell receptor is the entire antibody on the surface of the B cell. The T cell receptor So a T cell receptor is just basically this portion here of an antibody. So they both.
We're gonna hope this door's watertight.
Yeah. Okay. I was worried it was a tornado warning.
Yeah, then we're kind of do something.
Yeah. Sorry. To get back on track. So the B cell receptor is an antibody in its full form on the surface of a cell, whereas the T cell receptor is only the top portion of the variable region that is present on the T cell. So that's the T cell receptor. They're both antibodies, but it's different portions of the antibody. Does that make sense? Any questions? Yes. Basically, yeah. And what's critical is that the T cell receptor will bind peptide antigens that are displayed. So the B cell receptor binds to free antigen that's circulating around, whereas the T cell receptor will only see the peptide antigen that's been presented to it. We talked about antigen presentation, where one of the antigen presenting cells. So we have 3 different antigen presenting cells. We have APC for antigen presenting cell. You have B cells. You have macrophages and dendritic cells. So these are the 3 antigen presenting cells. What's important is that the dendritic cell is considered the professional antigen presenting cell. It'll go around and it has these long extensions, and across all these different extensions, they can interact with lots of different T cells because the T cell is smaller than the dendritic cell. And so the B cell or the B cell or dendritic cell will actually go up and present peptide to the T cell receptor. And so, as I mentioned earlier, the T cell receptor is also an antibody, and that makes it an immunoglobulin as well in a structural sense. But it's not the entire portion of an antibody. But similar to an antibody, it'll bind a very specific epitope. So again, T cell receptors will recognize a specific epitope, much like the B cell receptor. And it'll exist, it'll have incredible domain variability between T cell receptor, between T cell to T cell, because again, similar with the B cell, the B cell receptor will recognize only one epitope. So one B cell will recognize one epitope. Just like that, the T cell receptor will recognize one specific epitope that's been presented to it. And they're both the B cell receptor and the T cell receptor are on the membrane, but again, it's not the T cell receptor is not the entire antibody. And a T cell is only making one version of it, but it's littered across the surface of the cell. The T cells will use the T cell receptor to recognize the antigen, but again, it'll only recognize one specific epitope. And so the way it's presented to the T cell receptor, the antigen, is through what's called MHC, or major histocompatibility protein. So the way this works is the MHC. So what's important first is the T cell receptor I mentioned is an antibody, but only a portion of it. And so if it bears a striking resemblance to a full form IgG if you look at just the antigen binding domain. And so it's an antibody, but not the entire antibody. Does that make sense? All right. So MHC. MHC is major histocompatibility complex, and in humans, it's referred to as HLA, human leukocyte antigen. And MHC comes in two specific flavors. It comes in MHC class 1 and MHC class 2. So what I think I mentioned last lecture is that MHC class 1 is present on all nucleated cells. It's almost like an ID that says, I am Dan. And in the presence of viral infections, we talked about interferons being expressed, and interferons will increase the expression of MHC class 1. And so all your nucleated cells will be screaming, I am Dan. They're saying, I am self. But what's important is that MHC class 2 is different. It's only expressed on antigen-presenting cells, on this list of cells here. So these cells express a large amount of MHC class 2. Dendritic cells, macrophages, and B cells. Now, I mentioned last class that in immunology, we're always learning and we're always changing things. And so if you were to do a literature search of what cells express MHC class 2, you'll find several other cells that also express it. But they're not professional, they're not legitimate antigen-presenting cells. These are the main cells. And the dendritic cell in particular is considered your main professional antigen-presenting cell. And what's kind of funny is that the way we talk about it in immunology is that these cells are promiscuous. Not in the slutty sense, but what they mean by that is that these cells will go around and they'll present whatever's in their MHC class 1 or MHC class 2. And so constantly your cells are engulfing whatever they can get their hands on, digesting it, taking those peptides, and putting it on the surface of MHC class 1 or MHC class 2. And in this image, in this crystal structure image, you can see that there's peptides for MHC class 1 on the top right portion. There's a little gray peptide present there. In MHC class 2, it's between the blue and the yellow ribbons. And so they take these antigens, engulf them, chop them up and digest them, and then present them. And they're constantly doing that. And what will end up happening is they'll go around and promiscuously just go to cells and say, do you recognize this? Do you recognize this? Do you recognize this? Do you recognize this? And so for MHC class 2, what will end up happening is with dendritic cells, they have a really unique structure in that they have these like long extensions. And they express a lot of MHC class 2. And so it'll be all across these different extensions. And this is much larger than a T cell. In your pre-lecture, it said a T cell is on the order of, I think, 7 or 8 microns in diameter. A dendritic cell is on the order of 20 microns in diameter. And you'll have within the way that the dendritic cell has evolved is that to serve its function of promiscuously interacting with lots of different T cells, it'll be able to talk to different T cells at one time. And so within these little pockets, it'll be able to talk to lots of different T cells at one time. And so as I mentioned earlier, the peptides will fit into these little pockets that are expressed on the surface of your cells. And so MHC class 1 will present peptides and say, is this self? Is this not self, basically? And MHC class 2 will take things that it's ingested and saying, it'll be presenting it and saying, should we mount an immune response to this? And what's important is that with MHC class 1, that's on the left, it'll only present peptides on the order of 8 to 10 amino acids long. So with all of these peptides, these are on the order of, 1 to 10 amino acids that we're talking about. So these are really small structures. For MHC class 1, it's on the order of 8 to 10. For MHC class MHC class 1, so you can imagine a normal size hot dog, all right? So that's an 8 to 10 amino acid long hot dog. But for MHC class 2, it can present peptides on the order of 18 to 20, so nearly twice as long. And you can envision an 18 to 20 amino acid hot dog being presented on the surface of MHC class 2. So why do we care about major histocompatibility complexes? As I mentioned earlier, it's among the primary ways in which immune responses can be mounted. There are three main qualities with MHCs. The first is that they're polygenic. Each class of MHC is comprised of many different MHC genes on different chromosomes. And so it requires lots of different genes to create a specific copy of MHC. Next, it's polymorphic. There are many different alleles or variations with MHCs. When they're on different genes, when they involve different genes, there's the likelihood of there being polymorphisms that create different variants. And so within this room, we all likely, assuming there aren't any cousins or siblings in this room, we all will have likely different alleles of our MHC genes, and thus creating different MHC proteins on the surface of our cells. Lastly, they're codominant. What that means is that you make both copies that you get from your parents, both alleles that you receive from them. You're expressing both. So there's not one allele that's dominant over the other. They're both expressed together. And so what's critical about this is that any two people inheriting the same two alleles of MHC class 1, remember, class 1 is self. That's like your ID card, your cell's ID card, that it belongs inside you. And so in the context of organ transplantation, between these MHCs being polygenic, polymorphic, and codominant, it's incredibly rare for people who are not related to have the same blood type and MHC variants. And so you need for transplantation, MHC matching needs to be on the order of 90 to 95%. Because again, if that's not the case, your immune system will mount an incredible immune response to an entire organ, and it'll be catastrophic. And so if you're not related, and if you're not the same blood type, or if you're not related, it'll be hard for both blood type and MHC variants to be matching. Are there any questions? I guess, thank you. Yeah. Yeah. So as the matching blood type is not as hard to get. It's the MHC matching that makes it more difficult because there's such variation across between the different genes involved with MHC protein. Blood type versus MHC? MHC. Because blood type, there's only so many different kinds of blood types that we have, whereas there's so many different polymorphisms within MHC that creates so much variety. All right, so I'll stop the lecture there. And if you guys have any further questions, feel free to come on down and ask. Thanks a lot.